we've heard a little more about the background and goals for this project, let's talk to one of the scientists who was directly involved with the original research.
Hi, my name is Justin Meyer. I'm a professor at UC San Diego, an assistant professor. And how did I get into biology? As a little kid, I loved looking under rocks and finding lots of insects and different creepy crawly things, catching fish out of ponds and catching frogs and things like that. I love to capture them. I'm sure they did not love to be captured. And I love to look at them in jars and to look up in books how they what they ate, what habitats they lived in, how they were adapted to these different habitats. And then as a college student, that interest in just biodiversity led me to want to pursue biology. And then from there, I didn't know exactly what field of biology I wanted to go into, but I learned that evolutionary biology was a very vibrant field in biology, that Darwin hadn't answered all of our questions about how we get adaptations and how we get all of these really incredible diverse organisms in nature. And so I got interested from that early childhood curiosity. So I'm an assistant professor. I'm a principal investigator. That's also short. PI is short for principal investigator. And so you'll hear that as shorthand often. What does that mean? So at an institution like UC San Diego, your primary job is to run a research lab. You also teach, and as a part of running that research lab, you also mentor graduate students and postdocs, and sort of develop the next generation of researchers. And so your job is multifaceted. Every day I do something slightly different, whether it's teaching or mentoring or running statistics on people's data, or managing a lab, or there's just lots of different things that I have to do, or give talks, or write papers. So if you are interested in being a researcher, it is an amazing job and it's very fun, but it requires lots of different skills, and not just being good at math and science like I had thought when I was undergrad, but you also have to focus on writing and developing those skills and communication skills and different things like that. So I think this new mechanism for how viruses evolve host range expansions is very significant in that, well, it's new. Basically, it's a huge question in evolutionary biology, but also in virology and in the science of emerging diseases. The question is, how do viruses jump from one species to another species? So when you have an Ebola outbreak, that's usually a unique event where a virus has evolved a series of mutations that allow it to move from bats into the human population. HIV has jumped from chimpanzees to humans many times. You hear about the bird flu and the pig and swine flu, and those are all occasions where a virus moved from one organism into a completely new species. And so it's just a major question of how does that even work? So for a long time we've been able to identify what are some of the causal mutations that allow the virus to shift from one host to another host. But we haven't been able to really understand how they work. We can just identify them. So what this mechanism and what our research is on is really understanding, okay, not just identifying the mutations, but figuring out, what is the molecular mechanism? How does it exactly change the virus so that it can then jump to a new species? So now the mission really is to figure out how common is this new mechanism. Is this just some weird thing that we observed in a lab? There's data out there that suggests it's not just something that we observed and something that would happen in our virus, but actually that it's more general. And so that's the mission of this class. That's the mission of this project, is to figure out how general is this new mechanism. And so its relevance is to understanding how viruses jump between different species. Sure, we're studying it in phage, but it probably is relevant for human pathogens as well. If we find that this new mechanism for how viruses evolve to jump into new host species, if we find that it's very common, then what do we do with this information? So, I mean, certainly learning things to understand how the natural world works is useful, but, can we actually use this in a way, this information in a way to better deal with things like Ebola or other emerging diseases? And the answer is that, yeah, if we are able to, or if we find that this is a general mechanism, then there's kind of two ways that we can use that information. One way is that we can use it to help as sort of a fingerprint for viruses that we need to worry about. So if we're able to detect bistability in viruses, so that's what you guys are doing, then we can use those assays that you're performing, and maybe some other assays that we could design in order to detect whether or not there's other viruses, whether or not there's things like influenza that are infecting chickens, or other viruses that seem like they could jump into human populations. We could use this as a quick way, a very cheap and affordable way to actually assay those populations and determine whether or not they're at that sort of threshold where they almost have the right mutations and the right characteristics to jump into the human population. So I definitely think that it could be used for surveillance. But the other thing that we could do is that if these phages and other viruses that are in the midst of expanding their host range are unstable and have this bistability characteristic, then that's an Achilles heel. So these phages we've already shown, are more sensitive to temperature. They're more sensitive to proteinase K, which is an enzyme. It's a compound that can eat apart proteins and can be used against these viruses. And I'm sure there's lots of other ways that we can actually interfere with these unstable viruses. And so, yeah, so I think this information is important to learn, just to learn fundamental biology, but it does have these applications in that we can develop surveillance techniques, but also develop ways to intervene and stop the really potentially deadly viruses. I want to thank you all for taking part in this research. It's very important work. I think we're all going to discover new things that we didn't even anticipate. And I really look forward to seeing your results. We really could not do this kind of research without having all of these different students and classes working on this project. There's no way that me and my lab myself could accomplish this. And so I want to thank you all.
Thank you.